In the 1970s, amidst a world unraveling the secrets of DNA, one man stood on the cusp of revolutionizing genetics forever: Edwin Southern, an unassuming biochemist at the University of Edinburgh. Southern had one question: how do we pinpoint specific DNA sequences among the billions of letters in the human genome? The challenge was monumental, but Southern was relentless. In 1975, he unveiled a groundbreaking technique aptly named the Southern Blot. Using this method, scientists could isolate and identify specific DNA sequences with precision. It was as if Southern had handed humanity a magnifying glass to study the very blueprint of life.
The Southern Blot wasn't just a discovery, it was a revelation. It laid the foundation for modern molecular biology.
From identifying genetic mutations to diagnosing diseases, Southern's technique changed everything.
His innovation sparked countless breakthroughs, from cancer research to forensic science, paving the way for DNA fingerprinting and the Human Genome Project. Edwin Southern's discovery was more than a method, it was a beacon of hope and understanding, reminding us that even the smallest discoveries can illuminate the vast mysteries of life.